Transcription is something that happens in both prokaryotes and in eukaryotes. But in eukaryotes, making a messenger RNA that's ready to be used for making protein is a little bit more complex. So let's compare. So in prokaryotes, let me get my pointer. Let's say this is the gene for a protein, the protein ATP synthase. It catalyzes formation of ATP. That's the gene. And what happens in prokaryotes, the gene is copied by RNA polymerase into a messenger RNA, and the messenger RNA is used to make the protein ATP synthase. In eukaryotes, the gene that codes for ATP synthase is a little bit more complex. So how is it complex? Just imagine that the letters and words that I'm using in here is the signal on DNA, is the gene for this protein. And we know that the letters on DNA is A, G, C, and T, but I'm giving you English words in here just to make the point. So on prokaryotes, the gene for ATP synthase, it basically spells out ATP synthase. But in eukaryotes, the words and the phrase that is used to spell out that protein are interrupted with nonsense sequences. So instead of saying ATP synthase, you have A, blah, TP, blah, synth, blah, A's. So this blah is supposed to represent that blah, blah, the usual blah, blah we have in our everyday language. So the gene, the way it is, the sequence in the gene is actually quite nonsensical. And before you can make protein out of it, you have to get these nonsensical sequences out. So what are these nonsensical sequences called? They're called introns. And then the ones that, if you attach them, they would make sense, are called exons. So during transcription, first, a RNA molecule is made that still carries these nonsensical sequences, and we make this initial RNA, we name it primary transcript. And the primary transcript is not used for making DNA. First, we need to get these nonsensical sequences out. Once we get them out, then you have the final messenger RNA, which can then be used to make ATP synthase. Now, just this complexity makes transcription in eukaryotes a little bit more complex, but it also gives various different ways for eukaryotes to control transcription and also to add complexity to the proteins that they can make. But we'll talk about how that works when we talk about gene regulation. Okay. So what is the process that is involved for removing these nonsensical sequences? It's called RNA splicing. So this primary transcript is put through the reactions of RNA splicing, which is a form of RNA processing, and then the introns are out. Now, but before the RNA can leave the sequence, it also has to be further processed in eukaryotes. And there's two other things that happen. On the 5' end of the RNA, there's a series of guanine nucleotides that are added to the 5' end of the RNA. And on the 3' end, there is a series of adenine nucleotides added to the 3' end of the RNA, end over end. These are long poly A tails. We call them the poly A tail. So the RNA processing that happens in eukaryotes to the primary transcript is splicing plus addition of the 5' G cap and then the addition of poly A tail at the 3' end. Now, what is the function of this poly A tail and the G cap? They are involved in two different things. First of all, they prevent the RNA from being degraded. Now, there are enzymes sitting in the cell and they're just looking for floating around pieces of nucleic acid that don't have any protection, and they just chop them off. But then if you build protections like these, then you can protect the messenger RNA from being degraded. So one of the functions of the GCAP and the poly-A tail is protection from degradation. Another role is to help the messenger RNA to move out from the nucleus. Now, the nuclear membrane has a very strict gate that won't allow just anything to go in and out of the sequence. It has to be specific sequences associated or signals associated to the molecule that was to go in and out. So these sequences allow the messenger RNA to exit the nucleus.